DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of four chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. It means that wherever there is a T in one strand, there will be a in other opposite strand, and wherever there is a C, there will be a G in the opposite strand. Each strand has a 5' end and 3' end. The two strands run in the opposite directions. This determines how each strand of DNA is replicated. The first step of DNA replication is to separate the two strands. The unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called primer. This marks the starting point for the construction of the new strand of DNA. Enzyme called DNA polymerase bind to the primer and remake the new strand of DNA. DNA polymerase can only add DNA bases in one direction from the 5' end to the 3' end. One of the new strand of DNA, the leading strand. It's made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can formally make this strand in series of small chunks called Okazaki fragment. Each fragment starts with an RNA primer. And then DNA polymerase then adds a sorted DNA bases in the 5' to 3' directions. The next primer then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme called exonuclease reviews all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fill the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragment of DNA in both strands to form a continuous double strands. DNA replication is described as semi-conservative because each DNA molecule is made of one old conserved DNA and one new one.